Sit down, please. Thank you. I'm going to recommend that there are no bags on the table today, please. No bags on the tables. There are things, if you are wearing baggy clothing, like sweaters or sweatshirts that might knock into things, I recommend either tying them back. There are two over there, so that's Eric and that one. Nope.
Oh, now you tell us it's too late. We already have it. It's too late. We already have it. It's too late. Today.
Sierra, that's not going to work, please.
My mother's birthday is in like a couple of days, I think.
My mom's birthday is in like
I'm already 16.
Guys, starting off procedure A, what is our task today? What are we doing?
Being focused.
Prepare a wet cell
directed by your teacher.
Alright. So you guys see the list of stuff that is on here. This is giving you a protocol. You guys have done this before. This is review for the lab practicum. Remember the lab practicum where I had the table set up in different ways? There's all a timed rotation where you had to go through. You guys are going to have a final with that. I'm going to be providing you guys with guided questions and give you guys lots of topics that you need to know. So you don't waste your time. Like I won't waste your, right? I don't want to waste your time. Any questions right now pertaining to what is going to be happening? So I will give you guys that information later. Right now though, today, your task is to look at onion cells underneath the microscope. We've all done this. The skills I'm looking for you guys today are to perform, obviously, the onion dissection. So you're going to have to be able to pull up cells. You're going to have to show me individually that you are able to identify what a cell is under that. And then also you're being tested on your skill prep, your ability to make an actual physical slide. There's an additional factor that you guys did not do this last one. Last time, you guys just looked at the cells, you put it under low light to increase the contrast so you could see the parts. This time we're going to be adding a dye, a color, pigmentation, something that will actually change the color of all the stuff you're looking at that will make certain parts stand out even more so. So I gave a warning earlier that if you have like baggy sweatshirts or anything like that, or zippable sweatshirts, zip them up. Because if you have a sleeve or something like that you knock into, that stuff will stain. Just give you guys a heads up, it will stain. Any questions right now about that? Okay. So it's very important that you guys all through. There's also another warning. You guys obviously know the use of sharp tools in dissecting equipment. There are multiple razors and or scalpels available for you guys to use. The cutting station's right over here. This is the lab prep station. So everyone's going to come over here, cut up the onion section, and then make your slides over there. If you have any questions? Okay. One final note. We're obviously limited on microscopes, so some people may or may not be doing certain things. Go ahead in your questions, look at your stuff. Go in your notebook and look at your prior lab. Look at the lab that dealt with microscope slides and using onion cells. Use that information to kind of help answer some of the questions that are on here. So if you're not completely working on a microscope or you're unable to just because there's a limited number of them, use your time wisely. Stay on task with that. Any questions? Okay. You guys may get started. Again, supplies are all back there. So whoever gets up goes get to it. Cover slips over here. They're in the back as well. There's slides here. Sandy. Salute, salute. I have slide wipes. Onions are over there. Do you want to make that right here? You may. Can you maybe fill this up and tap on it, please? Go halfway. Thank you. Eric, how you doing? Good? Good? You guys waiting? good. You jump out of the way and get
How do you sprain a finger trying to read a book?
I had changed my clothes for work, right?
finish your story and then you get
Brand
it.
new shoes. I took my shoes up, and you know how you don't hit the bug right way, the bug will down and over the side of your shoe. So it was a high top bug, so I'm sitting there. So I said, alright, I didn't see the bug out, so I put my clothes on, and I had
I have something to make right now, so. No, you're taking the lead and making the slides. Alright, guys, you have to line up for Ben. Ben will help you. We'll put the slides going for you.
And you switch to book.
Oh, he did have it on. I was like, I don't know. I did that last
Awesome.
summer. Now he gonna bring it up every day.
Can I see your drawings? Thank you. Very nice.
She's taking you to Ariana. Kelly came. Kelly, yeah. You have to come back. I guess. Oh, go right here.
Just go to 4DX.
Come up.
So are you able to, do you understand? Could you be able to repeat that?
Mine look like bridges.
Yeah, so they look kind of long, right? Then were you listening to the description? Why is it different? Why do these look different from the last sentence?
Because we're looking at it from a different view.
Yeah, how can you tell?
Yeah. So I was saying, imagine looking at Kelly. If she's sitting here right now, Ben.
Yeah.
If you look at me, I'm looking at her standing this way. I see her straight up. She looks like she has a head, torso, legs, and whatnot. But if I were to change my perspective and look straight down on her, I would see her head and shoulders.
Oh, I know.
So what we first did with the onion cells, we were looking straight down on them. Now we're doing a side view of the onion cells. So we're changing it from circular pieces that we saw were kind of square pieces to longer rectangular pieces. In reality, it's a three-dimensional thing. It has shape, it has depth, it has length and whatnot. So it has volume to it. So we're just changing our perspective of where we're going. Okay.
Wait.
Yeah, how does that?
That's fine. You're in the maximum.
Huh? What question? Oh, he was talking at which one? Because we was doing number 2, where it says what shape are these cells?
Heard you afraid.
So guys, if you are
One.
on
Do
questions,
you see
I'm sorry, which
a whole
questions?
bunch of them,
Question
or do you
number
just see one big
4,
cell?
guys, just give me a heads up. Question number 4 talks about trying to estimate how many cells there are and actually figure out the size. The best measurement to do that is at 10x. Count the number of cells that are there, and then I'll help you guys out with the formula afterwards. So just, all you have to do is put 10x and just count the number of cells.
But on 10, I can't see anything.
Or is that not possible?
Hey, yo.
Just look all-
How much time do we have left in class?
We have about 10 minutes, so that's what I want to do is get us finished up now.
Alright.
So guys, let's start kind of going over these questions briefly. I'm going to choose and select which ones are more important. So obviously number 1, cells, skin of an onion. Many cells, 1 cell. Many, okay, let's keep it simple. What's the general shape?
Rectangular.
So remember that whole idea that the shape of that cell, if it looks rectangular, what did it initially look like when we first looked at cells? At the onion cells? It was kind of round. What's the difference between when we saw these round cells versus rectangular cells? What's going on?
Right, the rectangular cells form as a structure, like a clear structure.
These are the exact same cell. Why do they have different shape? Because that's a better way to ask.
Oh, yes.
We're viewing them at different perspectives. So if we were to take Aaron, have him stand up for a minute, stretching out.
Hey.
Hey, Aaron, if we look at Aaron this way, we see him as really tall, but if I were to take him and look down straight on top of him,
You
only
just see
has
his
a certain area.
shoulder.
So have a seat, thank you. This is tall Aaron, this is tall onion cell. This is short, small area of Aaron. This is just looking down on top of the cell.
Oh.
Do all these cells have cell walls?
No.
Yes. Why do they have cell walls?
Because it's a plant.
Because it's a plant. So you write that down too. If you don't know that, it's a plant. You should have that down. Go to the back side. What does the stain do?
Makes it more visual.
Enhances the image. That's, I think, the best word to use is enhance. What does it allow you to see? Cells outline.
In
Structures such as
Structures
cytoplasm, which looks like what?
and the nucleus. It's like a grainy.
Okay, looks grainy. And then the nucleus. What color is the nucleus? Stain. So any type of cell membrane with methylene blue actually gets stained. Any other organelle that has a membrane to it gets stained. That's why the nucleus has that. If you look very closely, you can see nucleolus, which is exactly where the DNA is located. Sierra had a really good one. I don't know, is it still set up? No, it's not. But she had a really good nucleus where you can see the actual center of the cell where the DNA was located. So if you still have a slide, kind of look around and see if you can find one of those. Generally, if you have your slide set up and you have your piece of onion skin underneath your thing, usually the edges are the best spots to actually see what has been affected by the dye. The dyes are stained, seeped in sideways. Good. Any other questions about this?
No, sir.
Okay. This again is review. It's going to become something part of your practical. We have 3 more classes left before you guys have that in class. So I'm going to be giving you guys other little mini labs. 1 DNA lab, just so you know. Keep this, just so you know this becomes part of the study sheets. Any questions? Cool. Be sure to throw out the slides, turn off the microscopes, cover them, push them towards the center. Keep your areas nice and clean. Thank you. Cool.
Oh.
Alright guys, what are our objectives today? Alright, so we're gonna look at onion skin. Excellent. How so? Peel it. Exactly, put glass in. Put water on it. Seal it right up. We're gonna check out and see if we can identify the parts. Our objectives today, you're gonna be able to perform an onion dissection and make a slide. I'm gonna ask you guys to document the use of methylene blue stain. You're gonna be using this specific stain for this class. We're going to look at the specific thing, the job of methylene blue and how it affects them. So I'm going to ask you guys to look at these cells beforehand and look at the cells afterwards. I'm going to just ask you guys to observe the differences between cells with dye and cells without the dye. Do you have any questions about that?
Nope.
You guys need a reminder about how to cut the cell, cut up the onions and such? Pardon? Okay, some people will help. And I have some people I know that will take the leadership too, but I'll help if you need it, absolutely. Any questions whatsoever? You can start right now. So we have supplies over here. I want to kind of keep this area open, have you guys use these microscopes over here. Alright? On the wrist. You go side to side with it. It hurt. Is it really?
I
Even
keep it
isolate.
like this. That's how I shoot like this.
Wait, are we supposed to do the gloves?
Yeah, but that's.
The rest was smooth.
Are we supposed to use gloves?
You do not need to use gloves with these. These are no biohazards. So, and also you're taking just the skin, so you're going to be.
I'm not activated right now, I'm stressed. I just put the ink water on. I'm just so aggravated.
Together.
Like this.
And then.
I'm gonna eat this.
Damien, I gotta get you caught up too.
Then for me, when after you brush it,
So, Jermaine, go on down.
let it sit on a bowl.
Is this what you're gonna do with that?
You can have shaped up.
Yep, and if you can cut it into little pieces, then put it on slides. There's a water squeeze bottle right over there. You can still go and they'll still be able to see if there was injury from the past. But I would recommend it because you're
You
not,
want X-ray.
that's not something that you want to go to
Where's the
emergency
slide?
for, because that's more expensive. You want to go to your
like-
general practice.
Right here, right here, right here. Oh wait, no.
I know,
Don't
okay.
get the food. What do you mean? Don't you got that?
I got a skin boyfriend.
Some people do.
State medical.
Yeah, some people do, some people don't. I would assume. So I would just say it's more expensive.
I got tighties at home.
All right, this right here. This is the one you jump on, please.
Beatrice, I got tighties at home.
I
got it. Well, actually, I'm a grandma's son.
Themselves, answer questions. They have letters. Okay. So.
I can't put it.
Oh, no, oh, no, you
Yes,
didn't.
right, actually doing them right now, so you can decorate your own screen. So you got paper right there, you got one, right? And I'm gonna ask you guys to use these computers, these microscopes up here, because everything's all plugged in over here.
Oh, this shit is blazing.
Alright?
It's like a tiny knife.
Well, what one thing?
You started it?
Yeah, don't worry about it, just do
You heard me? You started
you're
it?
going to super long one, yeah, don't do that, just stick with this, or this one.
You started it? Huh?
You still can't see it. You see that blue.
No cuss words.
Too much?
On what?
I'll just leave this. See where it's down.
You have a blue marker, I mean crayon?
Yeah.
No, oh, never mind, I have that.
Now, Moss.
That's weird, why is it not focusing? That's a low power. Right there. What happened?
Oh, that looks cool.
Right? It looked like a waterfall or something.
Like dark all around.
Mm-hmm.
Oh my god, you're nuclear now. Really well.
Time for my special drawing.
Oh, wait, I'm not done yet.
It's okay.
I don't know how to answer
What up, bro?
number 6 and number 10.
How you doing?
Can you answer number 6 and number 10?
Good, good.
What's up?
Can you still see the double layer? Can you see like, let me see.
Hello? Hey.
Sort of, you can. With number 6, a double layer. Sort of.
So, stop it.
What happened? So why was it saying that? To see what?
Said what?
What did you get to see?
Everything.
Oh my god.
I read it. Good.
Oh my god, that is
Hey guys,
Oh,
escort
whip up,
cleanup,
whip up.
please. Hear me out. Clap once, can you hear me? Clap twice. Awesome. And now everyone's paying attention now. These slides, I don't want you guys to clean them off because it gets stained on yourself. So all you have to do is just throw them away when you're all set.
Anyhow, this is.
We are going to go over the questions once everyone cleans up. Okay?
Okay.
And then we will move from there. Good.
I guess my mother all the time. She gets so mad.
Starting now?
What?
Starting now?
Oh, okay. So what I'm doing is like I'm analyzing the amount of red, green, and blue light in each picture. So like I'm gonna open up all of these, and then I'm going to stack them to show like since these pictures look almost exactly the same, it's going to be like a time lapse thing. So then I'm going to find out like the change. Like a change in much like
Cool. I'm going to show a picture of what you're doing. So he's using a software called ImageJ. It's from the National Institute of Health. And it's a really kind of rough piece of software. You know how like if you use Photoshop, it's pretty easy to use. There's a lot of user guides and stuff like that. This is not so user friendly. So what's unfortunate is he has to take a lot of time just kind of looking at how to do these types of photo analysis. So he's looking at each picture and identifying the number of pixels that are red, that are green, that are blue. And he's going to track changes in the color of these corals over time. It's pretty cool. So now he's opening all these pictures and making what we call a stack. So he can ease up his time and do less work.
less work.
Yes, somehow. Right? It's kind of funny. Pretty cool.
Hey Steve, come and get this one.
Yeah, welcome to Sleeping Giant State Park. And as you guys are quiet, please. We're at Sleeping Giant State Park. You're gonna notice one thing. Wow, it's be heard. There aren't a lot of people here, so that's another good thing for us. Sleeping Giant State Park was established in Hampton. It was actually first accompanied by people who just want to settle and have vacation homes here. Their homes are on the upper side of the ridge. We are going to be doing 2 trips here. The first one is so we can do a full assay or full survey of the stream that's down below. And we're going to then go back to like what we did at West Rock Park, look for the source of water. The next trip is going to be involving a lot of hiking, so we're going to ask that you guys bring shoes that you can hike in. People have boots and stuff like that, that's great, it's going to help you out with the water. Water temperatures is around 62 degrees, 63 because of the rain. So that's something to think about for what we're doing and whatnot. So whether or not you want to get wet, it is going to get warmer today, so don't really worry too much about things. Our goal today is need you guys to work in your groups, collect individual data. There are clipboards still in the blue crate, as you guys know. Okay? And then there's also sampling materials. So you guys have nets, and with that big trawling net, instead of dragging it across the river, I'm going to have you guys do something different. I'm going to have people station themselves so they lock the net in place. There's a strong flow in here, unlike what was at East Rock Park. The strong water flow actually pushes things downstream and automatically slams it into the net, so it's easier for you guys to catch things. The things that we're going to look for are aquatic insects. He actually opened up the crate and passed out some of the clipboards.
We caught some of those bugs that were all crammed up together.
Yeah, exactly. There's a lot of insects on the bottom of rocks. And the bottom of these rocks, we're going to use these insects to actually identify the quality of water. Certain species identified will determine how clean the water is or how dirty it is. And I'm going to give you a preemptive warning already. The water here is extremely clean. This is why I make it our last trip.
This is our last trip?
This is our second to last trip. This is our last location. Why? So we have gone from very polluted waters to much cleaner, more pristine waters.
Nice one.
You guys make sure you have that. Now the reason I wanted to pass this out, I'm going to grab one of these. On the back side, you guys had these sheets. Some people have them, some people don't. I have extra copies in there. On the back side, there is a guide, girls. There is a guide to insects that you find, that you can find in streams here. This will help you identify. So you're going to find things that look similar to the insects that are here. Any questions about that? No? Alright guys, we're going to grab all of our stuff, we're going to bring it right down to the river. Alright?
Where's my pen? One of those papers?
Grab them.
Any more of these papers?
Yeah, they're more in the paper that down.
We going down? You gotta bring everything you're at.
No.
So Michelle, you gonna be on mine?
Yeah, I got one here.
You said.
Can someone help Nikki, please? Water samples for each group. You know how we did the lake, and you guys did top, middle, bottom, right? And you did a couple feet in between? Yeah. The difference is now I want to show you guys what the differences are in oxygen quantities between the ruffled area, or the riffle area, sorry, the area that has lots of bubbles. So that's called a riffle. That's an area that's going to have, do you think, higher or lower oxygen? Higher. There's more energy. Okay, good. There's more energy. It's turning the water. That's what you guys have for your hypothesis. You're thinking about, all right, there's a lot more oxygen. When you get out this way, where it's calm, what's going to happen to the oxygen? What's going to happen to the oxygen? It's going to decrease. All right, it's going to decrease. Awesome. That's what I want you guys to say or show through your water sampling. So I want 3 water samples from each group. 1 from up in the riffle, 1 from somewhere in the middle, and then 1 from down here. Got it? You can't walk through that shit, right? You're going to walk right through it. Sorry.
That's why you have on boots.
Yeah, that's why you have boots. Some people are going out there. That's fine. You're going to get wet. You guys know that.
Oh yeah, this is my time break.
Okay?
Oh, you're trying to be funny.
You got it good? Alright guys, begin. Grab some nets. Start exploring.
Who else in our group? What's the group when it's digging? I'll take it.
Yeah, put it on Instagram.
Aaron, are you gonna do water quality stuff up there?
Yeah, well, that's what we're gonna do. But...
Alright, you're gonna drop the. Alright, can you do above the waterfall and then below the waterfall, please? Thank you. We'll get it, don't worry. Read it? Okay. So it's loading.
Wow, wait a minute. Yeah, you really do have magic fingers because it's turned on.
Remember it has a loose cord so you get as a short.
Get in the water. Get in the water. In there.
Alright.
One, breathing. Two, it's freezing, Victor. You need to go in. Yes, I did touch the water. It's freezing, I know.
It's only 62, guys, right? Well, you can tell us once you drop that in. Language. Alright, what do we have for readings? They'll tell you.
874. 8.74. Wait, I want to do something.
Can't see with the reflection. Here we are.
The temperature. Where's the temperature? Oh,
58 degrees.
Yep.
It's 58 degrees, Gabe.
58?
Yep.
That's bad.
Just be careful. Well, you're already wet anyways.
That's bad and cold.
What are you doing?
Getting ready to go in.
Oh, alright.
Slowly.
Slowly.
Where's the total?
We were doing that with a paper test. So we're not doing that here. And we can't do turbidity because it's not a lake. Exactly, it's not a lake. We do have them, but it's use that in lakes. And about, you have to estimate your depth.
Estimate?
Yeah.
Oh, you don't,
How deep? You don't need to. I mean, just like, how deep do you think this water is right here?
A foot and a half.
Exactly, exactly. You're 100% right.
Yeah,
I got some fish. Oh, you got a lot of them. Look at you.
That was easy.
Can I take a picture?
Yes.
Good job. Now we're going to ID them. That's a lot. So these are all little larval fish, or little babies.
Alright, ready? Come on.
You can see they're not so happy though. It was a little abusive to them. Alright, you guys going to try out here? Yes! This area is even better.
You guys check out the Daphnia. It's pretty cool. You can see where its little babies are located. You can see its heart beating. It's pretty cool.
Let me see. That's perfect.
So can you show them where the heart's located?
Oh, Dave. Did you see the eggs?
Oh yeah, the Trichoptera.
Yeah, put it in
Oh yeah, guys. Look at this guy.
Put it with the snake. The snake does. Oh, you can't see the heart beating. Yo,
Take a look.
Sam.
Check
You
out
can see the heart beating like this on that shit, and you can see the eggs.
Check out the Trichoptera. That's pretty cool.
Let me see. Let me see.
Let
Let me see.
That one is a Trichoptera.
And which one? It looks like the one there, right?
Oh, yours?
Okay, no, look.
Look, just look at it. You can see the heart beating.
It's okay. Get a little extra water in there. Oh, you got it. Critter too. Nice.
Oh, I see it. Alright.